So how do we identify prokaryotes? How do we tell different species from each other? Well, it's pretty difficult, actually. And it's kind of, this slide's about Eubacteria, but it's pretty much the same for Arcobacteria. Under a microscope, we need a microscope to see individual prokaryotes. We can see colonies of bacteria growing in a petri dish, but we can't see the individuals that make up that colony. So they're very, very tiny. We have to use a microscope, whether it be a light microscope or an electron microscope. It's impossible to identify different species visually. In other words, even if we use a microscope, for example, this electron micrograph of these rod-shaped bacteria, known as bacilli, there are many species that have that same shape. So just looking at their shape is not going to help us identify them. Just looking at them under a microscope isn't going to help us identify them. So again, these are rod-shaped bacilli. We've got spherical cocci, and then we've got spiral-shaped spirilla. So that one is pretty easy to remember. These other two, you're just going to have to memorize. So microbiologists use differences in cell wall structure, and really the ultrastructure. In other words, what makes up the cell wall, metabolism. In other words, how energy and matter is handled by different species of bacteria to identify different species. And we also use this term strain, because even within species, for example, the E. coli that lives inside our digestive system, there are different strains of E. coli that live in the digestive systems, even different strains within humans, but there are certainly different strains living within, for example, a cow versus a human. So looking at living eubacteria using a light microscope can help distinguish shape and locomotion, because if you're using a light microscope, you can look at living bacteria, and if they are able to move around, if they have flagella and are able to swim, for example, you'll be able to see that locomotion while looking at them under the light microscope. You're not going to even be able to see that, though, under an electron microscope. But under an electron microscope, you'd be able to better see the flagella themselves. So even though you wouldn't be able to see them moving, because remember, an electron microscope, you can't look at living cells. Still might be able to tell that they have locomotion because of their flagella. So in these all three of these are scanning electron micrographs, and in these we really can't see anything other than shape. So the best way to tell them apart is to alter their nutrient supply. In other words, grow them in petri dishes with different food sources, and then see what kind of waste products they give off. That's really the best way that's been developed to be able to tell them apart. We can now tell them apart too, however, using looking at their DNA sequence. We can sequence their DNA and tell different species apart by sequencing their DNA, and that's the ultimate way to tell them apart today. But the very first thing, like if you go into the doctor and you have a sore throat, the doctor is probably going to swab your throat and culture that bacteria to try to determine why you have a sore throat, what bacteria is living in your throat. So they get a sample from your throat, they put it on nutrient media in a petri dish, they grow that bacteria, and then they stain it. They might do what's known as a gram stain. So this process is known as gram staining, and it's simply fixation just means getting the bacteria to stick to a slide, a slide for a microscope. Then you add crystal violet stain, which stains the bacteria dark purple, and then you use iodine to make sure that dark purple, or to it reacts along with that dark purple to add another level of stain, basically, and fix it or cause the stain to really stick to the peptidoglycan in the cell wall, because that's really what it's sticking to, or staining. And then decolorize it, and they usually use alcohol to do that, or always use alcohol to do that, and that washes away the stain, but not of the gram-positive bacteria, because that stain has really stained the cell wall. But in gram-negative bacteria, there's a membrane around the cell wall that keeps the stain from staining the cell wall. And then because these bacteria don't have any stain anymore, they wouldn't be visible under the microscope, so they need to be counterstained. So pretty much a third stain, if you count the iodine as a stain, a third stain is added to stain those bacteria where the crystal violet has been washed away by the alcohol, by the decolorization process. So we end up with gram-positive bacteria that are purple and gram-negative bacteria that are pink. So this determines the ultrastructure of the cell wall, because again, the difference is that the gram-negative bacteria have a membrane, an extra outer membrane around the cell wall. Gram-positive don't. So again, this information will be really useful to doctors in trying to determine what antibiotic to use to get rid of your sore throat. Because different antibiotics are going to work on killing gram-positive bacteria versus gram-negative bacteria. So that's really useful information. But the most effective means of distinguishing bacterial species other than DNA is metabolism. And that takes time, unfortunately. So again, you have to culture the bacteria. You have to sample it, culture it, and then gram stain it or give it various food sources and see what the waste products are. And all of that takes time because you have to culture them. So of all the species of organisms on this planet, as a group, prokaryotes have the greatest metabolic diversity. They can get their matter and energy in ways that us plants and animals and protists can't. So the two general requirements to sustain life, matter and energy. And when we're talking about matter, we're talking primarily about carbon. So, for example, your body is made up of organic compounds which contain carbon, are carbon-containing compounds, and that's what builds your body. We're talking about carbohydrates and proteins and nucleic acids and lipids. They are all organic compounds, and that's what makes up every living organism on the planet. So it's all carbon-based. So that's the matter we're really talking about. Metabolism, as I've explained numerous times previously, refers to all the chemical reactions that are taking place in a living organism, necessary to handle or process energy and matter. It's all about the chemical reactions that process or handle the matter and energy in a living thing. The two matter handling modes that we find in prokaryote metabolism, but also in eukaryote metabolism, is heterotrophic versus autotrophic. Heterotrophic, which literally translates to other feeding, and autotrophic, which translates to self-feeding. The two energy handling modes are cellular respiration, and that's breaking down food to get the chemical energy out of food, and photosynthesis, which is using the energy of sunlight to make food. So we can mix and match these two modes, the matter handling modes and the energy handling modes, to describe the different metabolic, the different ways that prokaryotes carry out their metabolism. So chemoheterotroph should be very familiar to you because you are a chemoheterotroph. You get your chemical energy from food, you get your carbon from food, so both the energy and the carbon's coming from food. And that's what you do. And that's what some bacteria do too, in other words, some prokaryotes. Photoheterotrophs, and the only examples that I know of that are photoheterotrophic are bacteria, get their carbon from food. So they take in carbon by bringing in carbon compounds, organic compounds from their surrounding environment, but they get their energy from light. So it's a strange combination that we don't see in any other organisms other than prokaryotes. Then we've got photoautotrophs, which are like plants. So that, again, the most familiar to you are photoautotrophs. They get their carbon from inorganic carbon dioxide from the air. And even though that's a carbon-containing compound, it's still considered inorganic, by organic chemists anyway. And they get their energy from light. And then we have chemoautotrophs. So notice that we've combined these two, these matter handling and energy handling modes in every way that we can here. And that's what prokaryotes do. So there are prokaryotes that do all four, that can do any one of these four things, and some of them can do more than one of these different metabolisms. But anyway, chemoautotrophs, only bacteria, get their carbon from inorganic carbon dioxide, just like photoautotrophs do, but they get their energy from inorganic chemicals, and this is what we refer to as chemosynthesis instead of photosynthesis. So these are chemosynthetic prokaryotes rather than photosynthetic like plants. And so an example of where this chemical energy can come from is hydrogen sulfide. And an example of that are the bacteria that are living inside the tissues. So this is a symbiotic relationship that bacteria have within the tissues of the tube worms that live around deep sea vents, where hot volcanic water is coming out of the ocean floor, and it contains a lot of hydrogen sulfide and other chemical compounds. And these bacteria, because there's no light down here at all, we're talking miles down, two and more miles down, these bacteria have to survive without light. This whole ecosystem has to survive without light, and the producers in this ecosystem are these chemoautotrophs carrying out chemosynthesis. So it's a really cool alien kind of environment. So we've got chemoheterotrophs like Mycobacterium tuberculosis. This is the organism that will live in your lungs if you have tuberculosis. Not a good disease to have. Then we've got cyanobacteria, which are photoautotrophs, just like plants are. And then we've got chemoautotrophs, which are also chemosynthetic organisms, like the bacteria that live around deep sea vents. The worms are not chemosynthetic. It's the bacteria that lives inside the worms that are chemosynthetic. So prokaryotic metabolic diversity is really high and variable. Is highly variable because they've been around so long. They've been around for 4 billion years, and they've had a long time to evolve. And as I've said before, life will find a way. If there's a niche to live in, then life will find a way to live in that niche. And that explains why prokaryotes are so metabolically diverse. They've been around the longest. And this last point, before leaving this slide, leads to the next slide. The two processes that chemoheterotrophic prokaryotes utilize to break down food and convert energy for cellular work is aerobic cellular respiration and fermentation. And we're going to take a look at that on the next slide. But note that aerobic means with oxygen, and fermentation is without oxygen. So aerobic cellular respiration is something that we're going to study in great detail in another unit, but what you need to know about it now, it's a catabolic process, catabolic meaning breakdown. So it's a process of breaking down molecules with oxygen. It requires oxygen to get the energy from those molecules. So we're talking about breaking down food molecules, and that's what you do. Your cells right now, the energy that they're using to keep you alive is coming from aerobic cellular respiration. It's being carried out in every mitochondria, in every cell of your body right now to keep those cells alive and keep you alive. Again, aerobic means with oxygen, or using oxygen, or the presence of oxygen. And anaerobic fermentation is without oxygen. Anaerobic is the opposite of aerobic. So it's the catabolic. And again, so again, it's breaking down, but it's without oxygen. So both of these are catabolic processes, breaking down processes of getting energy from food, but the difference is that fermentation is without oxygen. So anaerobic means without oxygen, or not using oxygen, or that there is no oxygen present. Obligate means you have to. There's no choice. You're obliged to. You have to. No option. Facultative means capable of, but not restricted to. In other words, you don't have to. Facultative is kind of the opposite of obligate. So you don't have to. In this scenario, or in this demonstration, there are cultures of bacteria. So these are 3 different cultures of bacteria. It's not the same culture over time. Okay, so that's important to understand here. A lot of times in these diagrams, we're looking at the same thing over time. In this case, we're looking at 3 different cultures of bacteria. And it's important to identify what's going on here. So we've got a liquid medium. Instead of like a solid agar in a petri dish, this is a liquid medium like broth that the bacteria are growing in. So we've got a test tube, we've got the culture medium, which is a liquid medium. We've got the air-medium interface. So this circle here in the diagram represents the transition from the media to the air above it. And because, and then we have the bacterial cells. So all the little dots are representing bacterial cells. And it's important to understand that at the air-medium interface, there's going to be a higher concentration of oxygen, because oxygen is going to dissolve into the liquid medium, and then it's going to diffuse. But diffusion is a very slow process, and the oxygen is never going to be able to diffuse all the way to the bottom, or the oxygen that does diffuse all the way to the bottom is not going to be very much, and it's going to be quickly depleted by any bacteria that might be in the bottom, if they are aerobic bacteria anyway, bacteria that require oxygen or can use oxygen for cellular respiration. So that means that down here at the bottom, there's a very low oxygen concentration or no oxygen. So that's really the crux of this entire demonstration. So you need to understand the concentration gradient here that we have in oxygen, if you'll recall that term from when we were studying diffusion. So this demonstration suggests that these are different species or strains of bacteria, because again, these are 3 different cultures. And notice how the bacteria are behaving differently in response to the oxygen concentration gradient. So the bacteria in culture 1 are obligate aerobes. So this is how we use this terminology. This is how we put it together to describe how these bacteria are behaving differently. So these bacteria are obligate aerobes. They have to have oxygen, therefore they're hanging out up here where the oxygen concentration is high. They can't live without oxygen. They're obliged to use oxygen. They have to. In culture 2, they're obligate anaerobes. They can't live with oxygen. So, and it's strange to us because we breathe, we have to have oxygen. If we don't get oxygen, we die. But there are prokaryotes that don't like oxygen. And some of the first living things on the planet, some of the first prokaryotes to appear on Earth, appeared at a time when there was no oxygen in the atmosphere. For like 2 billion years or so, there wasn't any oxygen in the atmosphere. So they evolved to live without oxygen, and some of the ones that are still here today have to live without oxygen because they're descendants from those that appeared on the Earth back when there was no oxygen in the atmosphere. But there are still, oxygen-depleted environments on the planet, those niches that bacteria have evolved to live in. And then in culture 3, we've got facultative anaerobes that are able to live with or without oxygen. But notice that the way that they've drawn it in this diagram, there are more bacteria up here at the air-media interface than there are down here at the bottom where there is no oxygen. So even though these are facultative, they can live without oxygen, they can live with oxygen, they seem to prefer oxygen, and an explanation for that is that it's more efficient. Aerobic cellular respiration is much more efficient than anaerobic fermentation, and you'll see why when we study those processes in detail. So the bottom line is that the diversity of metabolism that we see in prokaryotes is vast compared to other organisms.